Imagine looking at a photograph of your family, but instead of seeing their faces right in the dead center of the image, there's just a gray, blurry shadow.
Yeah, complete blind spot.
Exactly. And you try to look closer, maybe shift your eyes a bit, but the shadow just follows your gaze. The edges of the photo, the background, the frame, those are perfectly clear. But the most important part, the center, is just gone.
It's a profound loss of independence. And for, millions of people over the age of 50, that creeping gray shadow isn't some hypothetical scenario. It is the defining feature of their everyday life.
Right. We are talking about dry age-related macular degeneration, or dry AMD. It's a disease that slowly and sadly irreversibly erases the center of your vision, and right now there is no cure.
None at all.
But today we are going on a mission to understand what looks like a massive leap forward. We're doing a deep dive into a groundbreaking 2026 paper from the journal Cell Death and Disease about a revolutionary mini retina model.
It really is a fascinating piece of bioengineering.
It is. So our goal today is to leave no stone unturned for you. We're going to break down exactly how this biological model works, decode the specific cellular mechanisms of this blindness, and, explain every assay and protein involved. Okay, let's unpack this.
Well, to cure any complex disease, you really have to be able to accurately recreate it in a controlled environment first. For decades, the scientific community has been trying to solve human blindness using animal models, but those models just fall fundamentally short of human biology. The overarching theme here is that understanding the pathology of AMD requires a distinctly human context.
So before we look at these futuristic mini retinas the researchers built, we should probably understand the biological battlefield, right? What is actually happening in the back of the eye during dry AMD?
So the retina operates as a highly specialized, interdependent ecosystem. At the core of this system are your photoreceptors, the rods, and the cones.
The rock stars of the eye.
Basically, yeah. These are the sensory neurons that physically capture light particles and convert them into the electrical signals your brain eventually translates into vision. They are the main event, the highly specialized machinery that makes sight possible.
But those rock stars don't function in a vacuum, do they? They require, a massive amount of metabolic support.
They absolutely do. And that support comes from a thin layer of cells sitting just beneath them called the retinal pigment epithelium, or RPE. You can think of the RPE as the essential support crew for the photoreceptors.
Okay, so what does the support crew actually do?
Well, the RPE filters nutrients from the blood, it feeds the photoreceptors, clears away their metabolic waste, and generally maintains the structural integrity of the retina. But in dry AMD, which, by the way, accounts for about 90% of all AMD cases, that RPE layer begins to degrade and die off.
And when the support crew collapses, the photoreceptors are just left stranded.
Exactly. They essentially starve or drown in their own waste, leading to their degradation. And because the macula, the center of the retina, has the highest concentration of these cells, that's where that gray shadow begins.
Which brings us to the historical bottleneck in AMD research, because scientists have traditionally relied on mice or isolated cultures of RPE cells to study this degradation.
Right, but evolutionary biology presents a major hurdle there. Mice are largely nocturnal. They don't even have a macula. Their vision is entirely different from ours.
Wow, so testing on them is kind of missing the point.
Exactly. And if you just study isolated RPE cultures in a dish, you completely miss the downstream effect, which is how the human photoreceptors themselves actually die. The field desperately needed a way to study human photoreceptors interacting with human RPE.
Enter human-induced pluripotent stem cells.
Yes, the game changer.
They took these cellular blank slates and coaxed them into growing into retinal organoids. We are talking about three-dimensional biological models of a human retina, but the timeline required to pull this off is just staggering to me. They grew these organoids in a specialized culture for 180 days.
Half a year.
Yeah, half a year. Managing a living biological model for that long introduces a ton of variables. Why wait so long? Why not 30 days?
Well, because in neurobiology, maturity dictates function. You cannot accurately model an aging-related neurodegenerative disease on embryonic or immature cells. Over those 180 days, the stem cells differentiate. They self-organize into specific layers, and crucially, they develop mature rod and cone photoreceptors.
So they are actually acting like adult cells by that point.
Precisely. By the 6-month mark, these cells have developed distinct inner and outer segments. They have formed the necessary synapses, and they actually express the opsin proteins required to sense light. They are functionally mimicking the complexity of an adult human retina.
That is wild. It's essentially like aging a fine wine or, slowly growing a biological microeye from scratch just to see how it breaks down.
It is a remarkable feat of tissue engineering. And once they had this healthy 180-day-old human microeye, the next step was to simulate the disease. They had to subject it to the right kind of stress.
Right. So just do a quick recap for you listening. Dry AMD destroys your central vision by killing off the RPC support crew and the photoreceptors. Since mice aren't humans, scientists managed to grow 180-day-old human mini retinas in a dish. Which leaves me with a question for you. If you could grow a miniature version of any organ to study your own health, which would it be? Think about that as we move on.
It's a great thought experiment.
So getting older is the biggest overall risk factor for AMD, obviously. But the researchers decided to focus on the biggest modifiable risk factor, which is smoking.
Yeah, the clinical data on that is striking.
It really is. Smoking can accelerate AMD progression by 2 to 5 times. So the researchers exposed these mature organoids to cigarette smoke extract.
They applied this extract at very specific concentrations. It was 500 and 750 micrograms per milliliter, and they did this for 48 hours to basically stress test the system.
Okay.
And to observe the resulting cellular damage, they utilized a newly developed technique called 3D ARQ, which stands for 3D Automated Reporter Quantification.
Now, in the past, analyzing a 3D tissue sample meant you had to, flash freeze it, slice it into microscopically thin cross-sections, stain each slice, and look at them individually under a microscope, right?
Oh, yeah. It's incredibly time-consuming, and honestly, you're essentially looking at dead tissue.
But the 3D ARQ system completely bypasses that.
It does. It allows for high-throughput, three-dimensional fluorescent scanning of the organoids while they are still whole and alive. They use specialized molecular tags that literally light up in response to specific types of cellular damage, allowing them to map the progression of the disease in real time across the entire structure of the mini retina.
I do want to push back on the methodology a little bit here, though, because cigarette smoke is a highly chaotic substance. It's a chemical soup. It contains thousands of different chemical compounds, many of which are toxic in completely different ways. If you are trying to isolate variables and understand precise cellular mechanisms, why introduce such a messy stressor? Why not just use a clean, simple chemical to cause the damage?
That is a very fair point, and it touches on a fundamental debate in experimental biology, reductionism versus real-world complexity. The researchers actually anticipated this exact critique.
Oh, they did?
Yeah, they ran parallel tests using pure defined chemicals like hydrogen peroxide and sodium iodate, which are standard laboratory tools for inducing oxidative stress. And those chemicals absolutely cause damage. But the reality is that AMD is not a single variable disease.
Right. It's not just one thing going wrong. It's a whole systemic failure.
Exactly. It's driven by a confluence of overlapping stressors. You have severe oxidative stress, lipid peroxidation, iron dysregulation, and mitochondrial dysfunction. The chemical messiness of the cigarette smoke extract actually mimics the complex reality of human pathophysiology better than a single sterile chemical ever could.
Oh, I see.
If we only ever test drugs against simple, isolated stressors in a lab, we risk developing treatments that fail the moment they enter the messy environment of the human body.
That makes total sense. So just to recap this section for you, smoking is the biggest modifiable risk factor for AMD. Researchers used cigarette smoke extract and a 3D scanner called 3D ARQ to trigger and measure realistic, complex damage in the mini retinas. So, question for you. Does knowing that a complex, messy stressor is better for lab testing change how you view standardized scientific models?
It definitely forces us to embrace the complexity. And once that multi-layered attack was underway, the researchers turned their attention to the exact mechanisms of cellular execution.
Yes, and here's where it gets really interesting, because cells in this context don't just abruptly stop working, right? When they are pushed past the point of no return, they trigger these complex, highly orchestrated death pathways. So exactly how are these cells dying?
Well, the researchers identified two distinct pathways happening simultaneously. The first is apoptosis.
Okay, the intrinsic pathway.
Right. Apoptosis is often referred to as programmed cell death. It is an internal self-destruct sequence, and it originates in the mitochondria. To observe this, the researchers used a specific fluorescent dye called JC1. It changes color based on the electrical charge of the mitochondrial membrane.
So when the mitochondria are healthy and maintaining their power, the JC1 dye emits a red signal. But if the mitochondria sustain too much damage and that membrane depolarizes, the dye shifts to a green signal.
Exactly. And in the smoke-treated organoids, The 3D scanners picked up a massive shift from red to green. The mitochondrial health was failing. This failure acts as an internal alarm bell, triggering a lethal cascade of protein interactions. Specifically, they tracked a sharp spike in a pro-apoptotic protein called BAD.
A bad protein, literally.
Quite literally, yes. And that spike happens alongside a simultaneous drop in a crucial survival protein called BCL2.
And the ratio between those two proteins basically determines if the cell lives or dies. When BAD goes up and BCL2 goes down, it tips the internal scales toward execution.
Yes, and that shifting ratio activates p53, which is a well-known tumor suppressor protein. In this context, p53 acts as the ultimate arbiter. When it determines that the damage is just too extensive to repair, it officially initiates the end stage by activating executioner enzymes known as caspase-9 and caspase-3.
The mechanics of it are almost architectural. You can think of apoptosis like a massive sinking ship. The mitochondria are the engine room. Once the engine room floods and loses power, the ship's captain, which is the p53 protein, makes the official call to scuttle the ship. The executioner caspases are essentially the crew following orders to open the bulkheads and deliberately sink the ship from the inside out so it doesn't damage the surrounding tissue.
That's a great mnemonic to visualize it. And the researchers were able to confirm this structural scuttling using other specialized markers. For example, they used PSV794.
What does that track?
So, healthy cells have a molecule called phosphatidylserine that stays strictly on the inside of the cell membrane. But as apoptosis progresses, the cell actively flips that molecule to the outside, like a biochemical distress flag. The PSV794 tag binds specifically to that flag. They also used T1L staining to detect fragmented DNA, confirming that the cells were literally dismantling their own genetic code.
But that internal self-destruct sequence was only half the story. The smoke extract also triggered a completely different, newly discovered death pathway called ferroptosis.
Right. Ferroptosis is a particularly devastating form of cell death driven by iron dependency. Using a dye called ferro-orange, the researchers observed a massive toxic accumulation of intracellular iron within the smoke-treated organoids.
And free iron inside a cell is highly reactive. It triggers something called the Fenton reaction, doesn't it?
That's the core of the problem. The Fenton reaction is a chemical process where that accumulated iron reacts to generate highly toxic hydroxyl radicals. These free radicals are incredibly unstable, and they immediately attack the lipids, or the fats, that form the protective outer membrane of the cell.
They initiate lipid peroxidation. The researchers were actually able to visualize this using a dye called BOBO-DIPY-C11, which shifts color the moment the fats in the membrane begin to oxidize. So if apoptosis is the coordinated internal scuttling of the ship, ferroptosis is more like the physical hull of the cell, literally rusting away from the outside in. The heavy metals build up, the oxidative stress spirals, and the structural walls rust away.
The resting analogy works perfectly for the cell membrane here. And normally a cell has mechanisms to fight off this rust. The ratio of reduced oxidized glutathione, or GSH to GSSG, acts as a critical antioxidant shield.
Right, glutathione is like the premier antioxidant.
It is. But the researchers found that during this attack, that GSH to GSSG ratio completely plummeted. The cell's natural antioxidant shield simply failed under the sheer volume of the reaction.
So to recap this intense cellular deep dive, the smoke extract kills photoreceptors via a dual threat. You have apoptosis, which is an internal self-destruction starting in the mitochondria, and ferroptosis, which is the iron-induced rusting of the cell membrane. Which leads to a question for you listening. Which mechanism sounds more insidious to you? The programmed internal self-destruct or the chemical rusting of the cell?
It really illustrates why this disease has been so impossible to cure. The photoreceptors are entirely outmatched.
They are outmatched, but identifying these executioners gives us the blueprint to fight back, which leads us to reading the proteomic analysis.
Right, they didn't just rely on visual fluorescent markers, they wanted to see the big picture. The researchers used liquid chromatography mass spectrometry, or LCMS, to analyze the entire landscape of proteins. They identified 159 differentially expressed proteins between the healthy and the dying organoids.
Okay, 159 is a lot. What are the key proteins we actually need to remember?
There are a few crucial ones. First, there was a severe downregulation in a protein called FOXO3. Now, FOXO3 normally functions as a guardian against aging and oxidative stress. But under the stress of the smoke extract, FOXO3 was essentially knocked offline.
Leaving the photoreceptors completely vulnerable.
Exactly. They also noted a collapse in a protein called CLIC4, which is vital for maintaining the physical architecture of the retinal layers. But while those protective proteins disappeared, others spiked dramatically, particularly HMOX1 and HMOX2.
And those are iron regulators, right?
Yes, the massive upregulation of those HMOX proteins is basically a biological signature of panic. It is the cell frantically trying to manage the severe iron overload that is driving the ferroptosis crisis.
The mass spectrometry data just perfectly aligns with the visual 3D scanning. But I do have to bring up a significant caveat from the paper here.
Let's hear it.
These 180-day organoids are amazing, but they're still isolated structures. They don't have a fovea, which is the center of the macula. They completely lack blood vessels, and most notably, they have no microglia, which are the immune cells of the central nervous system. Are we really curing AMD if we leave the immune system out of it?
Well, this raises an important question, and it highlights the ongoing evolution of bioengineering. No single model is perfect right now, but having a human photoreceptor model is still a massive leap.
How so if it's missing those pieces?
Consider the current clinical trials for dry AMD. A major focus right now is cell replacement therapy, trying to transplant new RPE cells into patients.
Okay, the support crew.
Right. But if the disease has already progressed to the point where the photoreceptors have died from apoptosis and ferroptosis, transplanting a new support crew is completely useless. The neurons they are supposed to support are already gone.
Oh, wow. Hadn't thought of that.
Exactly. So this model allows us to screen neuroprotective drugs that can keep the photoreceptors alive long enough for an RPE transplant to actually work. In fact, they use this platform to test a specific peptide called H105A and demonstrated it could actively block these death pathways.
That makes total sense. So to recap the final piece of the puzzle, proteomics revealed exactly which proteins malfunction, like FOXO3 going offline. And while the model lacks blood vessels and immune cells, it is the crucial missing puzzle piece for testing neuroprotective drugs in humans. So, question for you. How do you think researchers will combine these mini organs with artificial immune systems in the future?
That is the exact next frontier of this research.
So what does this all mean as a final takeaway?
It means that by meticulously mapping every assay, protein, and death pathway in a 180-day human retinal organoid, science has finally created a testing ground to stop the irreversible blindness of dry AMD. We have pulled the disease out of the body and mapped every vulnerability.
It is phenomenal. But it leaves me with one final provocative thought for you to ponder. We just spent this deep dive exploring how scientists grew a human mini retina to map the precise protein failures of a complex aging disease.
And the retina is an extension of the brain.
Exactly. So if we can achieve this level of precision for macular degeneration, what other incurable neurodegenerative diseases in the brain are just waiting to be mapped and cured in a petri dish? Are we going to see Alzheimer's or Parkinson's modeled exactly like this?
The methodologies refined here are highly translatable. The implications for the rest of the brain are genuinely staggering.
A new era of medicine. Well, thank you for taking this highly detailed journey with us today. Keep asking questions, keep digging deeper, and we'll catch you on the next deep dive.